everyone. My name is Kelsey Yu. I'm currently a junior attending Troy High School, and today I will be sharing my paper regarding the factors of osteosarcoma metastasis adaptation in the lungs. So before we begin, a little about me. I currently reside in Diamond Bar, California, and my interests include playing tennis, drawing, and listening to video game soundtracks. My career goal is to eventually become a medical oncologist and researcher, so I really wanted to focus this project on something on a cancer such as osteosarcoma so that I can learn a little bit more about the research process. So, cancer has long been regarded as a disease that is caused by the uncontrollable division of cells. It has been a widespread problem among the human population since even the earliest recorded records. Osteosarcoma is a variant of cancer. It exists as a primary malignant bone tumor that originates within bone, within these cells called osteoblasts, which are cells that create new bone. I first became interested in osteosarcoma due to its high incidence among the adolescent and young adult population, and I found that when it metastasizes, survival rates sharply plummet from 70% to 20%. And as one can see from this, when osteosarcoma or any other cancer reaches that state of metastasis, the patient's life is definitely put in a higher degree of danger. And I also found that lungs is most common location of metastasis. So that's how I kind of arrived at this topic. So what exactly is metastasis? Well, this can be explained through a process called the metastatic cascade, which is how the tumor cells move to one side of the body to a secondary site. So I'm going to be going over 3 steps, focusing on how exactly tumor cells are transported. So first, in intravasation, the tumor cells enter the vein so that they can be put into circulation in the bloodstream. Once these tumor cells are in the bloodstream, they can travel to the body to the metastatic site where they can exit the vein through a process called extravasation. If conditions permit, they can then possibly colonize the area for metastasis. Before I begin research, I find it important to specify a central hypothesis. So for this, I would say osteosarcoma tumor cells utilize both elements of the tumor microenvironment as well as their intrinsic properties in order to make alterations that facilitate further tumor expansion. For today's agenda, in terms of the adaptations, I'm going to be first going over the intrinsic metastatic potential, then the extracellular signaling molecules, then the role of cancer-associated fibroblasts, and end off with the extracellular matrix stiffness. The first factor I found to contribute to osteosarcoma metastasis is its tumor cells' intrinsic metastatic potential. Well, what makes up this potential? This can be explained through the oncogenes. The driver mutations, paired with the suppression of regulatory genes that normally protect the cell, allow for further cancer growth and dissemination. Paired with chromatin remodeling and changes in epigenetic regulation, this can culminate in something called oncogenesis, or the creation of new cancer cells. For some examples, studies have shown that the loss of a gene called PREX1 resulted in a negative impact on osteosarcoma proliferation, migration, as well as its capacity to form colonies. This points to the PREX1 gene to being a component and to be playing a role in osteosarcoma metastasis. Furthermore, the WNT pathway played a role in osteosarcoma expansion and metastasis as well. An increased tumor heterogeneity was shown to contribute because in a study, tumor cells that were exposed to the osteosarcoma tumor microenvironment actually evolved from being more phenotypically homogeneous or more uniform to increased tumor heterogeneity. And lastly, the MAPK pathway was able to increase the expression of a gene called MCL1, which actually inhibits apoptosis cell death. Another important factor to consider is the cell-to-cell communication that allows for possibly osteosarcoma gaining the upper hand in metastasis. CXCL14 is a chemokine in tumor communication, and it supported lung metastatic formation in this study by converting normal tissue-associated fibroblasts into metastasis tissue-associated fibroblasts. In this study, it was also shown that this molecule actually activated actomyosin contractility as well as matrix remodeling abilities. Thus, it really helped tumor environment to be more phenotypically supportive of metastasis. TRIM66 shown to act as an oncogene TGF-beta signaling pathway. Additionally, in the study, it was shown to downregulate tumor suppressor protein TP53 in osteosarcoma cells, which allow for the inhibition of cell apoptosis. TIM3 is another signaling molecule that was shown to facilitate a process known as epithelial to mesenchymal addition, which basically increased cell migration. So it was able to increase that volume of tumor cells that were coming in through the metastatic cascade. Additionally, it was also shown to active kappa beta / snail signaling pathway. So moving on, cancer-associated fibroblasts are a large component of the tumor microenvironment. They assist in creating a tumor-supporting phenotype. And a study showed that bone marrow stem cells that were exposed to osteosarcoma cells actually displayed possible indications of cancer-associated fibroblast presence. And with this in mind, these stem cells actually had a significantly improved proliferation as well as invasion ability. So thus, this study showed that cancer-associated fibroblasts was an important factor in altering the tumor microenvironment to be more supportive to metastasis, and it kind of boosted that process. And following that pattern of degradation, myofibroblast reprogramming was actually shown to eliminate fibromatrix degradation. And osteosarcoma cells that were incapable of forming tumor spheres were able to gain a tumor sphere forming ability among the addition of a protein called fibronectin. So lastly, the last adaptation I'm going to be going over today is extracellular matrix stiffness. The extracellular matrix is a network of proteins that is responsible for supporting cells in the body and providing them with regulatory environmental cues. The previously mentioned cancer-associated fibroblasts are able to cause the stiffening of the extracellular matrix or the hardening of the extracellular matrix proteins by adding on to the secondary site in the body. Eventually, this can culminate in a state called lung fibrosis. Once lung fibrosis occurs, it has been shown to significantly increase the rate of osteosarcoma metastatic formation in the lungs. And to add on to that, a study showed that there was an increased presence of YAP molecules in metastatic areas. For context, YAP molecules have been shown to facilitate metastasis, and this presence of YAP molecules actually decreased in a softer environment. This can then lead one to the possible conclusion that a stiffer environment through mechanisms such as ECM stiffness is needed for metastatic adaptation. Very interesting. So that being said, here I have illustrated a diagram of the various adaptations I have gone over in this presentation. So as one can see, during the metastatic cascade, those tumor cells go from primary tumor to the secondary site. In this case, it's the lungs, and the extracellular signaling molecules interact in their own ways with the tumor cells in order to support this process. And particularly, CXCL14 transforms resident fibroblasts into cancer-associated fibroblasts, which then build up ECM stiffness, which blocks out like a lot of the body's defenses, for example, the cytotoxic T cells. So this really shows how all the components in this metastasis process really work together in order to support the spread of cancer. So from this, I've come to the conclusion that in the process of metastasizing in the lungs, osteosarcoma tumor cells adapt to a variety of external and internal factors to survive. Tumor communication proves to be especially important in creating an immunosuppressive environment in order for the cancer to metastasize successfully. So what are some potential therapies? From my research, I have identified 4 that may be helpful in potentially treating osteosarcoma metastasis. So the first one is sorafenib, which is a medication that led to cell apoptosis and a reduced tumor volume. Next, nintedanib and pirfenidone are 2 therapies that both target that lung fibrosis. So these therapies really look to get rid of that tumor supporting phenotype. Here I have their 2 compounds in image form right on the slide. And lastly, AZD5991 is an inhibitor of the MCL1 gene. So basically, it's trying to restart that process of cell death again and getting rid of things that aren't supposed to be there. It's actually still undergoing testing and not yet FDA approved, but I saw it to be pretty interesting and something worth mentioning for this presentation. So lastly, what is needed in the field? Well, after studying multiple aspects of osteosarcoma metastasis, I found a study that showed that out of 75 osteosarcoma cases that were diagnosed with lung metastases, a state called lung calcification was observed in 47% of these patients. But when I tried to do further research on this lung calcification, I found little to no trials on it, and I feel that it could be a potential research subject for future researchers to consider when studying osteosarcoma metastasis. And of course, any of the genes or the extracellular signaling molecules I mentioned, for example, the PREX1 gene or the CXCL14, TRIM66, and TIM3 signaling molecules are all novel targets for future therapeutic opportunities to solve this very difficult disease. Here are my references for my information, and I would like to thank everyone for listening. I hope this presentation was enlightening.
Wonderful, wonderful, Kelsey. Now we're going to open it up for questions to the audience. So does anybody in the audience have any questions for Kelsey in her presentation? And if you want, if you're an attendee, you can just type it in the chat. That would be the best. Okay. We have one. What treatments are working best to stop these cancer cells from adapting, Kelsey?
Oh, yes. So, mostly from what I have seen, the treatments that have worked out the best are the ones that target these adaptations. But one specific thing I want to mention, because usually people think of chemotherapy when treating cancer, but I've actually found in research that osteosarcoma cells are very difficult to treat with radiation treatment. So, mostly it's these treatments that are more medication based instead of radiation based that work the
Awesome.
best for them.
Awesome. Thank you. We have another one. Are there any warning signs in patients that can tell doctors when cancer might spread to the lungs?
Ah, yes. So this usually happens when patients start exhibiting those starting symptoms, like they start feeling water in their lungs, or they may feel uncomfortable and difficulty breathing. And once X-rays are done, this can properly be classified as a lung metastases or lung fibrosis in this case. So once they already know that they have osteosarcoma and they start experiencing those symptoms in the lungs, they might know that, oh, well, it's probably metastasized at that point.
Awesome. Thank you. And one last one. What was the hardest part of your research process?
Yeah, actually, the hardest part of my research process was coming up with things that I felt like were more needed in the field because I feel like everything has been explored so well. But I kind of found
Mm.
trouble coming up with things that I felt like could be explored further. But luckily, I was able to do that. So it worked out pretty well.
Wonderful. Well, you did a fantastic job. Thank you so much, Kelsey.
Thank you.